How does an animal's niche affect its behavior?
A niche can be defined as the natural history of a species, where it lives, what it eats, and so forth. Those constraints in turn have an effect on behavior. For example, lizard species found in deserts from different parts of the world use a very similar behavior to harvest water from the moist air in the early morning. They use the bumps or ridges on their heads as condensing spots and then orient their bodies so that the water rolls toward the snout, where it can be licked off by the tongue.
Who was the first to postulate that microorganisms are the cause of disease?
Robert Koch, 1843 to 1910, was the first to identify that various microorganisms are the cause of disease. His four basic criteria of bacteriology, known as Koch's postulates, are still considered fundamental principles of bacteriology. Koch was awarded the Nobel Prize in Physiology or Medicine in 1905 for his research on tuberculosis.
Why do birds fly in formation?
Some birds, like geese and cranes, often fly in V formation because of the phenomenon of wingtip vortices. The lead bird in the V formation breaks up the wall of air that the flock flies into. The swirling air then helps push along the birds behind. However, being the lead bird is hard work, and the leader may then drop back to allow another bird to take on the work of the lead bird. The V formation also gives geese the ability to watch each other and communicate, via honking, about likely landing spots.
What are the components of the circulatory system?
The components of the circulatory system are vessels, heart, and blood. The three types of vessels in a closed circulatory system are arteries, capillaries, and veins. Arteries transport blood away from the heart to the various organs in the body. Veins return blood to the heart after it circulates through the body. Capillaries form an elaborate network of tiny vessels that convey blood between arteries and veins.
What determines the type of bond that forms between atoms?
The electron structure of an atom is the best predictor of its chemical behavior. Atoms with filled outer shells tend not to form bonds. However, those atoms with 1, 2, 6, or 7 electrons in the outer shell tend to become ions and form ionic bonds. Atoms with greater than 2 or less than 6 electrons tend to form covalent bonds.
Is lactose intolerance a genetic disorder?
Lactose intolerance is due to reduced activity of the enzyme lactase. Lactase hydrolyzes the lactose of mammalian milk into the two monosaccharides glucose and galactose. Lactose intolerance is very common in adults, as expression of the lactase enzyme begins to decrease in humans at about 2 years of age. Estimates of lactose intolerance in the U.S. population vary. Congenital lactase deficiency is a rare autosomal recessive disorder and results in no lactase production. This is in contrast to childhood and adult onset lactase deficiency, which is common and is inherited in an autosomal dominant pattern.
Do whales really talk to one another?
Whales produce low-frequency sounds that allow them to communicate across long distances. Recent research has found that among the fin whales, only males produce these calls. The long low-frequency sounds of male fin whales attract females to patches of food, where mating can then occur. This means that the increasing amounts of sonar activity from ships in the ocean may interfere with the ability of these males to find mates, and therefore threatens the species' survival.
What are some typical disorders of the immune system?
Allergies, autoimmune diseases, and immunodeficiency diseases are different kinds of disorders of the immune system. Allergies are abnormal sensitivities to a substance that is harmless to many other people. Common allergens include pollen, certain foods, cosmetics, medications, fungal spores, and insect venom. The antibody immunoglobulin E, IgE, is responsible for most allergic reactions. When exposed to an allergen, IgE antibodies attach themselves to mast cells or basophils. Mast cells are normal body cells that produce histamines and other chemicals. When exposed to the same allergen at a later time, the individual may experience an allergic response when the allergen binds to the antibodies attached to mast cells, causing the cells to release histamine and other inflammatory chemicals. While most allergic reactions are expressed as a runny nose, difficulty in breathing, skin rashes and eruptions, or intestinal discomfort, a severe allergic reaction results in anaphylactic shock. Autoimmune diseases are diseases in which the immune system rejects the body's own molecules. Insulin-dependent diabetes, rheumatoid arthritis, systemic lupus erythematosus, and rheumatic fever are autoimmune diseases. In contrast, in immunodeficiency diseases such as AIDS, the immune system is too weak to fight disease.
What is gradualism?
The Darwin-Wallace theory of evolution is based on gradualism, the idea that speciation occurs by the gradual accumulation of new traits. This would allow one species to gradually evolve into a different looking one over many generations, which is the scale of evolutionary time.
What are the functions of the circulatory system?
The primary function of the circulatory system is to transport oxygen and nutrients to all the cells of an organism. The circulatory system also transports wastes from the cells to waste disposal organs, such as the lungs for carbon dioxide and the kidneys for other metabolic wastes. In addition, the circulatory system plays a vital role in maintaining homeostasis.
What important fibers are obtained from the sclerenchyma of plants?
Many different types of commonly used fibers are obtained the sclerenchyma of plants.
What types of variations are there in plant stems?
Plant stems show variation in size and shape. In addition, some plants have modified stems. For example, the strawberry plant has stolons or runners, which are horizontal stems that grow along the surface of the ground. Iris plants also have horizontal stems called rhizomes. Rhizomes are large, brownish, root-like structures found just below the surface of the ground. The rhizomes store food and can spread to form new plants. White potato plants also have rhizomes. The rhizomes of the white potato plant end in large, round structures called tubers. The tubers are the part of the plant that we call potatoes and eat. Tendrils and twining shoots of the morning glory and sweet potato coil around objects and help support the plant.
What are the uses of chromatography?
Chromatography is used to separate and identify the chemicals in a mixture. It is useful to 1. Separate and identify the chemicals in a mixture. 2. Check the purity of a chemical product. 3. Identify impurities in a product. And 4. Purify a chemical product, on a laboratory or industrial scale.
Can unfertilized eggs function like other cells in the body?
A human egg is about 2,000 times the size of a sperm and has all the organelles and proteins required of a living cell, but only 1/2 of the necessary DNA. Because of this, the egg cannot be considered a functional cell. Without fertilization, human egg cells survive about 24 hours after ovulation. Prior to that, the egg is maintained by the support structures known as follicular cells. which transfer nutrients to the egg. However, in a process called parthenogenesis, an egg can develop without being fertilized. This type of fertilization occurs in rotifers, aphids, and whip-tailed lizards.
Why are mosses important?
Some mosses are decomposers that break down the substrata and release nutrients for the use of more complex plants. Mosses play an important role in controlling soil erosion. They perform this function by providing ground cover and absorbing water. Mosses are also indicators of air pollution. Under conditions of poor air quality, few mosses will exist. Peat is used as fuel to heat homes and generate electricity. Bryophytes are among the first organisms to invade areas that have been destroyed by a fire or volcanic eruption.
What are loofah sponges?
Loofahs are non-woody vines of the cucumber family. A fibrous skeleton lies inside of the fruit, and this structure is often used as a sponge. The term loofah is commonly used when this material is used as a sponge. Dishcloth gourd, rag gourd, and vegetable sponge are other popular names for this sponge.
How do dissecting microscopes differ from compound microscopes?
Compared to compound microscopes, dissecting microscopes, also called stereoscopic microscopes, provide a much larger working distance between the lens and stage in order to dissect and manipulate specimens. The light source on a dissecting microscope is above the specimen, since the specimen is often too thick to allow light to be transmitted from a light source below the specimen. Dissecting microscopes are always binocular, which provides a three-dimensional image.
What is bDNA?
There are three different structural forms of DNA. The secondary structure of DNA varies according to the amount of water surrounding the molecule. The structure that Watson and Crick described is bDNA. And evidence supports that this is the predominant form of most cells. bDNA is a beta helix with a clockwise spiral. Another structural form, aDNA, exists when there is less water immediately surrounding the molecule. aDNA is an alpha, right-handed, helix, but it is shorter and wider than bDNA. The third form, zDNA, is a left-handed helix in which the sugar phosphate groups zigzag. This form is found when certain specific base sequences are present.
What is the oldest genus of living trees?
The genus Ginkgo, commonly known as maidenhair trees, comprises the oldest living trees. This genus is native to China, where it has been cultivated for centuries. It has not been found in the wild, and it is likely that it would have become extinct had it not been cultivated. Fossils of 200-million-year-old ginkgos show that the modern-day ginkgo is nearly identical to its forerunner. As of the early 21st century, only one living species of ginkgo remains, Ginkgo biloba. The fleshy coverings of the seeds produced by females of the species G. biloba have a distinctly foul odor. Horticulturists prefer to cultivate the male plant from shoots to avoid the odor and mess created by the female tree.
How did the navel orange originate?
Navel oranges are oranges without seeds. In the early 19th century, an orange tree in a Brazilian orchard produced seedless fruit, although the rest of the trees in the orchard produced seeded oranges. This naturally occurring mutation gave rise to what we now refer to as the navel orange. A bud from the mutant tree was grafted onto another orange tree. Branches that resulted were then grafted onto other trees, soon creating orchards of navel orange trees. Every navel orange tree is derived from pieces of the tree that first produced the mutated fruit.
How can animals be grouped according to type of body cavity?
The structures of most animals develop from three embryonic tissue layers. The outer layer, the ectoderm, gives rise to the outer covering of the body and the nervous system. The inner layer, the endoderm, forms the lining of the digestive tube and other digestive organs. The middle layer, the mesoderm, gives rise to most other body structures, including muscles, skeletal structures, and the circulatory system. One system of grouping animals with three germ layers, triploblastic, is based on the presence and type of body cavity. Or chylome, a fluid-filled space between the body wall and digestive tube. Achylomates such as flatworms, phylum Platyhelminthes, have a solid body with no body cavity. Pseudocoelomates have a false chylome, since the body cavity is not completely lined with mesoderm. Pseudocoelomates include such animals as roundworms, phylum Nematoda, and Rotifers, phylum Rotifera. Coelomates, which have a body cavity that is completely lined with mesoderm, include most other animals ranging from earthworms, phylum Annelida, to vertebrates, phylum Chautata.
What is the cell theory?
The cell theory is the concept that as all living things are made up of essential units called cells, they are the fundamental components of all life. The cell is the simplest collection of matter that can live. There are diverse forms of life existing as single-celled organisms. More complex organisms, including plants and animals, are multicellular cooperatives composed of diverse specialized cells that could not survive for long on their own. All cells come from pre-existing cells and are related by division to earlier cells that have been modified in various ways during the long evolutionary history of life on Earth. Everything an organism does occurs fundamentally at the cellular level.
When was Dutch elm disease first identified in North America?
Dutch elm disease was first identified in 1930 in Cincinnati, Ohio. The source of the fungus was shown to be elm logs imported from Europe. By 1940 the disease had spread to 9 states. By 1950 it was found in 17 states and had spread into southern Canada. Today it is found wherever elm trees grow throughout North America.
What is X-ray crystallography?
X-ray crystallography, also called X-ray diffraction, is used to determine crystal structures by interpreting the diffraction patterns formed when X-rays are scattered by the electrons of atoms in crystalline solids. X-rays are sent through a crystal to reveal the 534 pattern in which the molecules and atoms contained within the crystal are arranged.
How is a gene library made?
To create a gene library, scientists extract the DNA of a specific organism and use restriction endonucleases to cut it. The scientists then insert the resulting fragments into vectors and make multiple copies of the fragments. The number of clones needed for a genomic library depends on the size of the genome and the size of the DNA fragments. Specific clones in the library are located using a DNA probe.
Do all animals have blood?
Some invertebrates, such as flatworms and cnidarians, lack a circulatory system that contains blood. These animals possess a clear, watery tissue that contains some phagocytic cells, a little protein, and a mixture of salt similar to seawater. Invertebrates with an open circulatory system have a fluid that is more complex. It is usually referred to as hemolymph, from the Greek term haimo, meaning blood, and the Latin term lympha, meaning water. Invertebrates with a closed circulatory system have blood that is contained within blood vessels. Vertebrates have blood composed of plasma and formed elements.
What is the most famous and deadliest of the puffer fishes?
Fugu gripes, the Japanese pufferfish, is a specialty in sushi restaurants. However, only specially trained chefs can safely prepare the fish for consumption. By minimizing the presence of tetrodotoxin, 1,000 times deadlier than cyanide.
Why are arthropods considered the most biologically successful phylum of animals?
Members of the phylum Arthropoda are characterized by jointed appendages and an exoskeleton of chitin. There are more than 1 million species of arthropods currently known to science. And many biologists believe there are millions more to be identified. Arthropods are the most biologically successful group of animals because they are the most diverse and live in a greater range of habitats than do the members of any other phylum of animals.
Who is considered the founder of modern conservation?
American naturalist John Muir, 1838 to 1914, is the father of conservation and the founder of the Sierra Club. He fought for the preservation of the Sierra Nevada in California and the creation of Yosemite National Park. He directed most of the Sierra Club's conservation efforts and was a lobbyist for the Antiquities Act, which prohibited the removal or destruction of structures of historic significance from federal lands. Another prominent influence was George Perkins Marsh, 1801 to 1882, a Vermont lawyer and scholar. His book Man and Nature emphasized the mistakes of past civilizations that resulted in destruction of natural resources. As the conservation movement swept through the country in the last 3 decades of the 19th century, a number of prominent citizens joined the efforts to conserve natural resources and to preserve wilderness areas. Writer John Burroughs, 1837 to 1921, Forester Gifford Pinchot, 1865 to 1946, botanist Charles Sprague Sargent, 1841 to 1927, and editor Robert Underwood Johnson, 1857 to 1937, were early advocates of conservation.
What were the contributions of Beadle and Tatum to genetics?
The work of George Beadle, 1903 to 1989, and Edward Tatum, 1909 to 1975. In the 1930s and 1940s demonstrated the relationship between genes and proteins. They grew the orange bread mold Neurospora on a specific growth medium. After exposing the mold to ultraviolet, UV, radiation, the mold was unable to grow on a medium unless it was supplemented with specific amino acids. The UV radiation caused a single gene mutation that led to the production of a mutant enzyme. This enzyme, in turn, caused the mold to exhibit a mutant phenotype. Beetle's and Tatum's work was important in demonstrating that genes control phenotypes through the action of proteins in metabolic pathways. In other words, one gene affects one enzyme. Since Beetle and Tatum were awarded their Nobel Prize in 1958, scientists have discovered that the relationship between genes and their proteins is much more complex.
What is a strain?
A strain or variety is a subcategory of a species. For example, Gregor Mendel's 1822–1884 work with garden peas involved various strains. One strain had purple flowers while another had white.
Can a cell survive in complete darkness?
Yes, cells can survive in complete darkness. A new theory on the origin of life argues that living systems arose in small compartments of total darkness located within iron sulfide rocks that were formed by hot springs on the seafloor. Cells of plant roots live in total darkness and carry out all normal plant cell activities, with the exclusion of photosynthesis. And, if you think about it, cells located in the very middle of your body also live in a pretty dark environment.
What is a centimorgan?
A centimorgan, cm, is a way of measuring the distances between genes on a chromosome. This unit was named in honor of Thomas H. Morgan, 1866–1945, who was one of the first to map genes onto chromosomes. Typically, a distance of a cm corresponds to a distance of about 1 million base pairs.
What is the accuracy of DNA fingerprinting?
The accuracy of DNA fingerprinting depends on the number of VNTR or STR loci that are used. At present, the FBI uses 13 STR loci in its profile, with the expected frequency of this profile to be less than 1 in 10 billion. As the number of loci analyzed increases, the probability of a random match becomes smaller.
How do cells eat?
Cells eat by a process known as phagocytosis, which is important to two types of cells: amoebas, unicellular protozoans, and mammalian white blood cells, macrophages, neutrophils. Phagocytosis is very important because it provides food for the protozoan. It also plays a critical role in defense systems by eliminating microorganisms or damaged cells in mammals. Once a particle, or microorganism, is ingested, It is wrapped within a vesicle, and then the vesicle fuses with the lysosome. The digestive enzymes of the lysosome then digest the contents of the vesicles.
What are effective and efficient types of pollination?
Effective pollination occurs when viable pollen is transferred to a plant's stigmas. Ovule-bearing organs, or ovules, seed precursors. Without pollination, there would be no fertilization. Since plants are immobile organisms, they usually need external agents to transport their pollen from where it is produced in the plant to where fertilization can occur. This situation produces cross-pollination, wherein one plant's pollen is moved by an agent to another plant's stigma. Some plants are able to self-pollinate and transfer their own pollen to their own stigmas. But of the two methods, cross-pollination seems more advantageous, for it allows new genetic material to be introduced. Cross-pollination agents include insects, wind, birds, mammals, and water. Many times flowers offer one or more rewards to attract these agents' sugary nectar. Oil, solid food bodies, perfume, a place to sleep, or sometimes the pollen itself. Other times the plant can trap the agent into transporting the pollen. Generally, plants use color and fragrances as attractants to lure these agents. For example, a few orchids use a combination of smell and color to mimic the female of certain species of bees and wasps so successfully that the corresponding males will attempt to mate with them. Through this process, pseudocopulation, the orchids achieve pollination. While some plants cater to a variety of agents, other plants are very selective and are pollinated by a single species of insect only. This extreme pollinator specificity tends to maintain the purity of a plant species. Plant structure can accommodate the type of agent used. For example, plants such as grasses and conifers, whose pollen is carried by the wind, tend to have a simple structure lacking petals, with freely exposed and branched stigmas. To catch airborne pollen and dangling anthers, pollen-producing parts, on long filaments. This type of anther allows the light round pollen to be easily caught by the wind. These plants are found in areas such as prairies and mountains, where insect agents are rare. In contrast, semi-enclosed, non-symmetrical, long-lived flowers such as irises, roses, and snapdragons have a landing platform and nectar in the flower base to accommodate insect agents such as the bee. The sticky, abundant pollen can easily become attached to the insect to be borne away to another flower.
Which children's author studied and drew illustrations of fungi?
Beatrix Potter, 1866 to 1943, perhaps best known for having written The Tale of Peter Rabbit. In 1902, began drawing and painting fungi in 1888. She eventually completed a collection of almost 300 detailed watercolors, which are now in the Armitt Library in Ambleside, England. In 1897 she prepared a scientific paper on the germination of Agaricinae spores for a meeting of the Linnean Society of London. Although her findings were originally rejected, experts now consider her ideas correct.
What are some famous figures in history with genetic disorders?
One of the most common genetic connective tissue disorders is Marfan syndrome. People with Marfan syndrome are usually very tall, with skeletal malformation and loose joints. Abraham Lincoln, Paganini, and Rachmaninoff were believed to have Marfan syndrome because of their above-average height and loose joints. People with Marfan syndrome may excel in sports such as basketball or volleyball. Olympic volleyball star Flo Hyman died of a ruptured aorta as a result of Marfan's. King George III, 1738–1820, of Great Britain suffered from porphyria, a disorder inherited as a dominant trait that causes occasional attacks of pain and dementia.
How many species of fungi are plant pathogens?
More than 8,000 species of fungi cause disease in plants. Most diseases found among both cultivated and wild plants are caused by fungi. Some pathogenic fungi grow and multiply in their host plants. Others grow and multiply on dead organic matter and host plants. Fungi that are pathogenic to plants can occur below the soil surface, at the soil surface, and throughout the body of a plant. Fungi are responsible for leaf spots, blights, rusts, smuts, mildews, cankers, scabs, fruit rots, galls, wilts, tree diebacks and declines, as well as root, stem, and seed rots.
How does kidney dialysis remove waste products from the body?
Damaged or diseased kidneys are not capable of removing toxic waste substances from the body. Kidney dialysis removes the nitrogenous waste and regulates the pH of the blood when the kidneys do not work. Blood is pumped from an artery through a series of tubes made of a permeable membrane and a dialysing solution. Urea and excess salts diffuse out of the blood as it circulates through the dialysing machine and are then discarded. Necessary ions diffuse from the dialysing solution back into the blood. The cleansed blood is then returned to the body.
What is herbal medicine?
Herbal medicine treats disease and promotes health using plant materials. For centuries, herbal medicine was the primary method of administering medically active compounds.
How are drugs detoxified by cells?
Drugs are detoxified by the smooth endoplasmic reticulum of liver cells. Detoxification usually involves changing the molecular structure of a toxin. A modification that increases the toxin's solubility, allowing it to be safely carried away by the blood and excreted via urine. Cells are able to increase their detoxification efforts when drug levels increase. Investigations conducted with rats that have been injected with a sedative known as phenobarbital have shown a striking increase in the amount of smooth endoplasmic reticulum of the liver cells.
What are the upper and lower shell of a turtle called?
The turtle, order Testudinas, uses its shell as a protective device. The upper shell is called the dorsal carapace and the lower shell is called the ventral plastron. The shell sections are referred to as the scuettes. The carapace and the plastron are joined at the sides.
What is the structure of a virus?
Viruses consist of strands of the genetic material nucleic acid. The basis of a genome, which is surrounded by a protein coat called a capsid. The capsid protects the genome and gives the virus its shape. Viruses may be either helical or icosahedral. Some viruses display a combination of helical and icosahedral symmetry, known as complex symmetry. The capsid is often subdivided into individual protein subunits called capsomers. The organization of the capsomers yields the symmetry of the virus. Animal viruses often form an envelope around the capsid. This envelope is rich in proteins, lipids, and glycoprotein molecules.
What do the numbers inside the recycling symbol on plastic containers mean?
The Society of the Plastics Industry developed a voluntary coding system for plastic containers to assist recyclers in sorting plastic containers. The symbol is designed to be imprinted on the bottom of the plastic containers. The numerical code appears inside a three-sided triangular arrow. A guide to what the numbers mean is listed below. The most commonly recycled plastics are polyethylene terephthalate, PET, and high-density polyethylene, HDPE.
How long does it take the average spider to weave a complete web?
The average orb weaver spider takes 30 to 60 minutes to completely spin its web. These species of spiders, order Araneae, use silk to capture their food in a variety of ways. Ranging from the simple tripwires used by large bird-eating spiders to the complicated and beautiful webs spun by orb spiders. Some species produce funnel-shaped webs, and other communities of spiders build communal webs. A completed web features several spokes leading from the initial structure. The number and nature of the spokes depend on the species. The spider replaces any damaged threads by gathering up the thread in front of it and producing a new one behind it. The orb web must be replaced every few days because it loses its stickiness and its ability to entrap food.
Why do animals defend territories?
Animals defend territories to protect their assets such as food, water, or mates. Animals will also defend the area where their offspring live. Because in evolutionary terms offspring are the ultimate asset.
How long can an orange tree produce oranges?
An average orange tree will produce fruit for 50 years, but 80 years of productivity is not uncommon. And a few trees are known to be still producing fruit after more than a century. An orange tree may attain a height of 20 feet 6.1 meters, but some trees are as much as 30 feet 9.1 meters high. Orange trees grow well in a variety of soils but prefer subtropical settings.
What is DNA supercoiling?
When DNA is not being replicated or specific genes are not transcribed. Its normal form is two strands twisted around a helical axis, much like a spiral staircase turning clockwise. However, during DNA replication or transcription, enzymes alter the structure of DNA such that additional twists are added. Positive supercoiling, or subtracted, negative supercoiling. Either type of supercoiling makes DNA even more compact. A group of enzymes, the topoisomerases, are able to disentangle DNA strands. They are called topoisomerases because they control the topology of DNA.
What are the shortest and longest chromosomes?
Among human chromosomes, the length ranges from 50 to 250 million base pairs. The longest chromosome is chromosome 1, with 300 million bases, approximately 10% of the human genome. And the shortest is chromosome 21, with 50 million bases.
What are the components of a gene?
The term gene describes a section of DNA that will be used as a template to build a strand of RNA or protein. In addition to this information, each gene also contains a promoter region, which indicates where the coding information actually begins, and a terminator, which delineates the end of the gene.
How does the mark recapture method estimate a wildlife population?
Researchers set traps to capture a population sampling in a given area. The captured animals are marked, or tagged, and then released. After a certain amount of time, the traps are set again. The second time, both marked and unmarked animals will be captured. The proportion of marked to unmarked animals gives an estimate of the size of the entire population. The equation used to estimate the number of individuals in the population is marked individuals x total catch second time n equals recaptured marked individuals, where n is the number of individuals in the population. A disadvantage of this method is that it assumes that a marked individual has the same chance of being trapped as an unmarked individual. Individuals that were trapped once may be wary of traps, or they may seek traps since they have learned that traps provide food.
What is the electromagnetic spectrum?
The electromagnetic spectrum is the range of wavelengths. It ranges from gamma rays with very short wavelengths, 10 to 5, and high energy through radio waves with longer wavelengths, 103 meters, and less energy. Visible light, seen as color, occurs between 380 and 750. Ultraviolet light has a shorter wavelength, while infrared light has a greater wavelength.
What is pasteurization?
Pasteurization is the process of heating liquids, such as milk, to destroy microorganisms that can cause spoilage and disease. This process was developed by Louis Pasteur, 1822 to 1895. As a method to control the microbial contamination of wine. Pasteurization is commonly used to kill pathogenic bacteria, such as Mycobacterium, Brucella, Salmonella, and Streptococcus, common to milk and other beverages. There are 3 methods for pasteurizing milk. In the first method, low temperature holding, LTH, milk is heated to 145 degrees Fahrenheit 62.8 degrees Celsius for 30 minutes. In the second method, high temperature short time, HTST, milk is exposed to a temperature of 161 degrees Fahrenheit 71.7 degrees Celsius for 15 seconds. This technique is also known as flash pasteurization. The most recent method allows milk to be treated at 286 degrees Fahrenheit 141 degrees Celsius for 2 seconds. This approach is referred to as ultra-high temperature, UHT, processing. Shorter-term processing results in improved flavor and extended product shelf life.
Can mushrooms grow up overnight?
A mushroom is only the fruiting body, that is, reproductive structure of a much larger fungus body that grows unseen in rotting logs, rich humus, and dark, damp places. Many familiar mushrooms have fruiting bodies that are fleshy and umbrella-shaped. Warm, damp weather triggers their sudden appearance. Usually first to be noticed are small, round button caps composed of densely packed hyphae. Soon after the outer covering ruptures, the stem elongates, and the cap enlarges to its full size. This entire process can indeed happen overnight.
When was DNA first discovered?
DNA was first discovered in 1869 by Johann Friedrich Miescher, 1844 to 1895. He isolated and described what he called nuclein, which eventually became known as deoxyribonucleic acid, DNA, the genetic information of the cell.
Why is Archaeopteryx important?
Archaeopteryx is the first known bird. It had true feathers that provided insulation and allowed this animal to form scoops with its wings for catching prey.
What freshwater mammal is venomous?
The male duck-billed platypus, Ornithorhynchus anatinus, has venomous spurs located on its hind legs. When threatened, the animal will drive the spurs into the skin of a potential enemy, inflicting a painful sting. The venom this action releases is relatively mild and generally not harmful to humans.
What disease is caused by protists of the genus Trypanosoma?
Species of Trypanosoma are the cause of sleeping sickness, East Coast fever, and Chagas' disease. All of these diseases are common in tropical areas. The trypanosomes that cause these diseases are transmitted by biting insects, such as the tsetse fly.
What are some economically important angiosperms?
Angiosperms produce lumber, ornamental plants, and a variety of foods.
What is polymerase chain reaction?
Polymerase chain reaction, or PCR, is a laboratory technique that amplifies or copies any piece of DNA very quickly without using cells. The DNA is incubated in a test tube with a special kind of DNA polymerase, a supply of nucleotides, and short pieces of synthetic, single-strand DNA that serve as primers for DNA synthesis. With automation, PCR can make billions of copies of a particular segment of DNA in a few hours. Each cycle of the PCR procedure takes only about 5 minutes. At the end of the cycle, the DNA segment, even one with hundreds of base pairs, has been doubled. A PCR machine repeats the cycle over and over. PCR is much faster than the days it takes to clone a piece of DNA by making a recombinant plasmid and letting it replicate within bacteria. PCR was developed by the biochemist Kary Mullis, 1944. In 1983 at Cetus Corporation, a California biotechnology firm. In 1993 Mullis, along with Michael Smith, 1932, won the Nobel Prize in Chemistry for development of PCR. DNA amplification is a method by which a small piece of DNA is copied thousands of times using PCR. DNA amplification is used in cloning, to detect small amounts of DNA in a sample, and to distinguish different DNA samples, as in DNA fingerprinting.
What do the numbers on a bag of fertilizer indicate?
The three numbers, such as 15-20-15, refer to the percentages by weight of macronutrients found in the fertilizer. The first number stands for nitrogen, the second for phosphorus, and the third for potassium. In order to determine the actual amount of each element in the fertilizer, multiply the percentage by the fertilizer's total weight in pounds. For example, in a 50-pound bag of 15-20-15, there are 7.5 pounds of nitrogen, 10 pounds of phosphorus, and 7.5 pounds of potassium. The remaining pounds are filler.
What is a control group?
A control group is the experimental group tested without changing the variable. For example, to determine the effect of temperature on seed germination. One group of seeds may be heated to a certain temperature. The researcher will then compare the percent of seeds in this group that germinate and the time it takes them to germinate to another group of seeds, the control group, that have not been heated. All other variables, such as light and water, will remain the same for each group.
Why do termites march around a ring made of ink?
Termite soldiers and workers are blind and so use pheromones to navigate. Two chemical compounds have already been identified as termite pheromones. It seems likely that the chemical formulation of certain inks contains compounds that mimic these naturally produced signals.
What factors affect enzyme function?
Enzymes may be controlled at a variety of levels, since proteins are coated with DNA. A change in DNA will change the rate at which an enzyme is produced. Control molecules known as competitive inhibitors may prevent the substrate from reaching the enzyme. Allosteric or non-competitive regulators may bind elsewhere on the enzyme, causing its shape to change and thereby altering its active site. Changes in environmental conditions such as pH, temperature, or salt concentration also affect shape. Absence of required enzyme helpers such as vitamins and minerals.
What criteria are used to identify emerging infectious diseases?
Emerging infectious diseases are ones that are new, changing, have shown an increase in incidence, or have the potential to increase in the near future. There are several criteria used to identify an emerging infectious disease, including 1. A disease causing symptoms that are clearly different from all other diseases. 2. Diagnostic techniques that allow the identification of a new pathogen. 3. A locally defined disease that becomes widespread. 4. A rare disease that increases in occurrence and becomes common. 5. A mild disease that becomes severe. 6. A slowly developing disease that increases in lifespan.
What cheeses are associated with fungi?
The unique flavor of cheeses such as Roquefort, Camembert, and Brie is produced by members of the genus Penicillium. Roquefort is often referred to as the king of cheeses. It is one of the oldest and best-known cheeses in the world. This blue cheese has been enjoyed since Roman times and was a favorite of Charlemagne, King of the Franks and Emperor of the Holy Roman Empire, 742 to 814. Roquefort is made from sheep's milk that has been exposed to the mold. Penicillium roqueforti and aged for 3 months or more in the limestone caverns of Mount Comelu, near the village of Roquefort in southwestern France. This is the only place true Roquefort can be aged. It has a creamy, rich texture and a flavor that is simultaneously pungent, piquant, and salty. It has a creamy white interior marked by blue veins. The cheese is held together with a snowy white rind. True Roquefort is authenticated by the presence of a red sheep on the emblem present on the cheese's wrapper. Penicillium camemberti gives camembert and brie cheeses their special qualities. Napoleon is said to have christened camembert cheese with its name. Supposedly the name comes from the Norman village where a farmer's wife first served it to Napoleon. This cheese is formed of cow's milk and has a white, downy rind and a smooth, creamy interior. When perfectly ripe and served at room temperature, the cheese should ooze thickly. Although brie is made in many places, brie from the region of the same name, east of Paris, is considered one of the world's finest cheeses by connoisseurs. Similar to Camembert, it has a white, surface-ripened rind and smooth, buttery interior.
How does carbon monoxide affect cell function?
Carbon monoxide is a highly poisonous gas. Because of its molecular similarity to oxygen, hemoglobin can bind to carbon monoxide instead of oxygen. And this subsequently disrupts hemoglobin's efficiency as an oxygen carrier. Carbon monoxide actually has a much greater affinity, about 300 times more, for hemoglobin than oxygen. When carbon monoxide replaces oxygen, this causes cell respiration to stop, leading to death. The particular danger of carbon monoxide poisoning lies in the fact that a person exposed to high levels of this toxin cannot be saved by being transporting to an environment free of the poison and rich with oxygen. Since the hemoglobin remains blocked, artificial respiration with overpressurized pure oxygen must first be performed to return the hemoglobin to its original function and the body to normal cell respiration.
Which animals are most demanding as pets?
There are several ways to answer this question. Physically, animals with very precise feeding requirements could be considered most demanding. An example would be the type of live food that a snake requires. On the other hand, animals with high learning capacity and communication skills can be quite exhausting for the owner. For many people, the physical and/or mental challenges of keeping a high maintenance pet like a chimpanzee or tiger can become overwhelming. Which is why so many exotic pets wind up being abandoned or given up for adoption.
How much cholesterol is needed in your diet?
None. The body is capable of making all the cholesterol it needs. Almost all cells can synthesize some cholesterol, but most require more to maintain plasma membranes. This additional cholesterol, which is found in the body, is synthesized by the liver. All cell membranes contain cholesterol, and it is also used in producing bile salts and steroid hormones.
What are some diseases associated with inadequate dietary intake of protein?
There are several diseases associated with insufficient protein intake. Marasmus, a condition caused by chronic malnutrition. Occurs most often among children 6 to 18 months of age. The arms and legs of children with marasmus are described as having a matchstick-like appearance. Kwashiorkor, another disease caused by malnutrition, derives its name from a Ghanaian phrase that describes the change a firstborn child often undergoes when a second child is born. Kwashiorkor sufferers typically consume enough calories but are protein deficient, usually a result of being weaned at too early an age. Kwashiorkor causes an enlargement of the liver and a buildup of fluid within the body, particularly in the belly. Children with kwashiorkor tend to have plump-looking limbs and distended bellies. But the deceptive enlargement is caused by swelling, not body fat.
Where do cancer cells come from?
When the reproductive rate of cells exceeds their death rate, the tissue becomes enlarged, forming a tumor. Although these cells are initially identical to the others in the tissue, they gradually take on characteristics of malignancy. The cancer cells reproduce rapidly and tend to be abnormally large or small. Malignant tumors grow 78 very quickly and invade other tissues. Cancer types are named for the location of the tissue that gives rise to the tumor and the organs involved. Genetics, viruses, or even environmental exposure to substances like those in cigarette smoke may cause tumor formation. However, not all tumors are malignant. Tumors that grow within a well-defined capsule are benign and unlikely to be life-threatening.
Which bear lives in a tropical rainforest?
The Malayan sun bear, Ursus malayanus, is one of the rarest animals in the tropical forests of Sumatra, the Malay Peninsula, Borneo, Burma, Thailand, and southern China. The smallest bear species, with a length of 3.3 to 4.6 feet, 1 to 1.4 m, and weighing 60 to 143 pounds, 27 to 65 kilograms, it has a strong, stocky body. Against its black, short fur it has a characteristic orange-yellow colored crescent across its chest, which according to legend represents the rising sun. With powerful paws having long, curved claws to help it climb trees in the dense forests, it is an expert tree climber. The sun bear tears at tree bark to expose insects, larvae, and the nests of bees and termites. Fruit, coconut palms, and small rodents are also part of its diet. Sleeping and sunbathing during the day, it is active at night. Unusually shy and retiring, cautious, and intelligent. The sun bear is declining in population as its native forests are being destroyed.